And we are back. So everyone, now we invite you to a unique experience. Now we are gonna have, we hope that you enjoyed the networking that you had around. So now you're gonna have the academia lab. We have two amazing presentations. Just let me call Kathleen to the stage. Let me check, invite her here. So today we're gonna have two amazing presentations from two researchers that are working on alternative proteins. Please help me welcome Christophe Chantre, co-founder and CEO of Boston Meats. This company is working on creating alternative meats with the right taste and texture. His research was carried out between the University of Zurich and Harvard University. We are also being joined by Dr. Kathleen Hefferon, author, lecturer, research scientist at Cornell University, a microbiologist with over 20 years of experience in plant research, and founder and CEO of Forte Proteins, an early-stage startup. Our host today is Nick Johnson, BME, PhD research student at UC Davis and president of the Davis Alternative Protein Project. So everyone, thank you for taking the time. Thank you for being here. Nick, please take it from me.
Hi, Anna. Thanks for the introduction. And thank you to ProteinX just for inviting me back to host another section of the Academia Lab. Today, I'm excited to talk with Christophe and Kathleen and hear about the innovations that they've come up with, both in research and with their companies. And so let's kick it off with Christophe. He's going to talk a little bit about some of the research he did during his postdoc, and then he's going to talk about the transition into creating a new company, Boston Meats. Christophe, you want to take it from here?
Yes, awesome. Can you hear me?
Yep, sounds good.
Awesome. So I'll share my, let's see if I can share my screen. Okay, I'll just share my entire screen. Do you see my screen?
Yes.
Yeah.
Awesome. Great. So my name is Christophe Chantre, and I'm the co-founder and CEO of Boston Meats. And so I'll give you a little bit of background on what we're doing, and I'll talk about how we came about starting this company and where we come from in academia. And so we're a company that's really interested in solving the problem of texture. So right now, as you might know, you have on the plant-based side, you have burgers, sausages, chicken nuggets. What you don't have are, chicken breasts, steaks, even something like pulled pork, where you really have this kind of distinctive muscle kind of structure. And that's really what we're interested in solving at Boston Meats. So for background, I met all of my co-founders in academia at Harvard back in 2013. And so we all essentially at the same time joined a lab, Kit Parker's lab at Harvard, and started working together ever since. Kit Parker's lab is a lab that's very interested in essentially how tissues grow, how they structure themselves, how you go from a couple cells to, a structured piece of tissue, how that structure then is going to inform its function. So if we're talking about muscle, it means, how can we create this fibrous aligned structure, and how that actually is going to influence how it's going to function, which is really its ability to contract. And so we kind of realized along the way that this structure function relationships that we were developing and understanding kind of roughly translated in the context of food to texture, to mouthfeel. And that's really what we were accordingly interested in developing. And that's why we kind of started Boston Meats around that. And I'll talk a little bit more about kind of what type of research we were specifically doing and I was really doing. But yeah, very briefly, so Boston Meats, so we formed the company a year ago, so we did a lot of research before launching this company. We have now 10 employees, and we're kind of continuously hiring. So check out our website if you're interested in talking with us. We've raised 1.5 million so far. We're planning on raising a lot more later this year. We're also the recipient of an NSF, non-dilutive NSF, so government grant. we're not really sure if there are any other kind of companies in the space that have received government funding for cultured meat research. But so if there are, that's great. If not, well, we might be the first one, which we're very excited about. And I think it goes to show that this landscape in this industry is really changing. When I started, there was really very little funding for this type of work, and now it seems to be changing more and more. As Nick, our host here, is recipient of an NSF grant as well in the context of academic research. We're also selected as a semifinalist in the X Prize competition, which is this competition where the goal is in the next couple of years to actually develop a real piece of meat that's made out of plants or cells, but not an animal. And really, it's supposed to have the same kind of sensory experience, nutritional profile. And so we're part of a couple of these teams that are part of this, and yeah, we're part of some other accelerator programs. But so, kind of for background, where I come from, so I have a training in mechanical engineering. I was really interested in, with this mechanical engineering training, go into work related to renewable energies or something related to climate, but kind of got interested in kind of cell biology along the way, and had the opportunity to join Kit Parker's lab at Harvard in 2013 with the goal of actually working on cultured meat. And so I joined this lab at a time where he had just secured some funding from the Tomcat Foundation for doing some initial research on growing meat, outside of an animal. And really, the goal there was more so than seeing this as a sustainable way for feeding humanity. It was the pitch then that he had made was we could create foods that are designer foods that have a certain health attribute, where you could add certain drugs or certain ingredients that are going to make it healthier for a specific reason. But so I joined this lab. I started working on actually growing 3-dimensional tissues using technologies that we developed in this lab. As I mentioned, Kit's lab was really specialized in understanding how tissues grow, and then from that understanding, developing tools to be able to regrow tissues outside of a human being or outside of an animal. And a lot of the work was geared towards medical applications, regenerative medicine, disease modeling, but he'd received this funding for doing some initial research in the context of food. And so I was able to join this lab and start working on essentially cultured meat, so growing 3-dimensional skeletal muscle tissues using the scaffolding technologies that had been developed in this lab. And so I did that for 8 months. It was extremely interesting research, and it kind of fit well with my original and personal interests in, related to sustainability and climate, and with the kind of the current expertise that I had, which was around bioengineering. And so I decided to stay on for a PhD. Unfortunately, there wasn't any kind of further funding for doing more research on cultured meat, and so we'd received funding, for work in regenerative medicine. And so I got really interested in skin regeneration, among other organs that we'd worked on. But one of the key organs that I worked on during my PhD was around skin, and understanding how skin regenerates, and in particular, why newborns have an ability to regenerate their skin a lot better than adults. And this is just some images here, essentially showing, for example, in utero, where surgeons perform operations in utero for one reason or another, whether it's a malformation, some sort of cancer indication, and they go and perform incisions and operations. They realize that by the time the baby is usually born, that baby does not have any scars at all, even if there are severe operations that were performed on that fetus. And so that led to a lot of studies in the decades that followed, including my PhD, which was really on understanding why that was, why certain tissues can regenerate better than others. And really what my work and a lot of the work from other people in this field kind of really came down to is that there's really structure and biochemical compositions that were very different and that really enabled to influence and support regeneration of those tissues. And so this was really work that was specific for wound healing, human health, but generally I think it informed a lot of the work that we did in the lab around choosing the right structures and the right kind of biochemical cues to be able to grow specific tissues, whether we're talking about muscle or fat in the context, or connective tissue in the context of meat, but a lot of other organ tissues. Now, so that was the work that I did, but our lab worked on a lot of different things. One of the kind of crazier and wacky things that came out of our lab a couple of years ago was this concept of creating a cyborg stingray. And again, it's really about this kind of this structure-function relationship. The goal of this project was to understand how a stingray swam, and it has this kind of very kind of particular way of, or undulatory way of moving its fins, which allows it to kind of move forward. And the goal of this project was to recreate that kind of that specific wave, and show that if you created that specific kind of structure or organization, you could actually make it swim in the same way. And that's what essentially was happening in that lab, and showed that, with the postdoc at the time that was leading this project, using light, he could control these little swimmers, which were the size of, of a coin, essentially. They were swinging them around in petri dishes in our lab, which was a lot of fun. And yeah, we worked on a lot of other projects around tissue engineering, heart ventricles, heart valves, and it was all around this kind of this structure-function relationship. How can we design a better structure so that it can have the function, the biological function that we're looking for? And so, in the context of meat, so obviously, if you're trying to recreate that actual experience of eating a piece of meat, well, we want to recreate, its core structure. And we kind of quickly realized that, if we wanted to create that accurate experience, well, recreating this kind of this fibrous structure that you have in a piece of meat is going to be really critical for, convincing more people to eat meat, because it's really going to impart, the texture, the mouthfeel, the juiciness, and your overall appearance of a piece of meat. And we kind of realized that there hadn't been a lot of work, or enough work that's done on recreating this kind of this fibrous structure that you have in a piece of meat. And that's really what we were interested in tackling in the context of cultured meat, and potentially starting a company. And so that's what we did. And so we published kind of on the side from our kind of core funded projects in regenerative medicine. We published this paper called Muscle Tissue Engineering and Fibrous Gelatin. And so it was leveraging our scaffolding technologies that we developed in the lab, and showing that if we could use these kind of fibrous scaffolding or these fibrous scaffolds, we could grow more structured kind of cell cultured meat. And that was, to our understanding, one of the first kind of scientific publications that came out showing that you could actually potentially do that and do it in a better way. And it obviously generated a lot of press from the New York Times and other outlets. But also, I think very interestingly for us, it generated a lot of interest from some of the large corporations out there. And so they were starting to ask us, can you apply this to cultured meat, but could you also apply this, to plant-based products? I think we knew this, but getting kind of confirmation from these large companies that, there was interest in, exploring our know-how and our technology in the context of plant-based as well, was really interesting for us. And I think it just, because, right now, again, as I mentioned, you have burgers, sausages, but you don't have anything like a chicken breast or a steak right now. And so that was, I think, for us, publishing this paper and then kind of having all these discussions was really strong motivation for us to kind of really move forward and launch this company. And so that's what we're doing right now. So we formed this company a year ago. And then, as I mentioned, we're really interested in developing these kind of more structured products and really essentially naming a whole new category of alternative meats that, in our opinion, doesn't really exist today on the market. And it's doing that using these novel technologies that we've developed at Harvard, and all the know-how over the years that comes from that research, that academic research, and now that we're translating into Boston Meats. And yeah, I'll stop there. It's been a very fun ride so far, and we're looking forward to next steps. And yeah, this is just a brief picture of our team members. Not everyone is full-time at this point, but that's a brief overview. And I'll stop sharing my screen.
Thanks, Christophe. That was awesome. It was really nice to see some of the things your lab is working on, both on the regenerative medicine side and that paper on cultivated meat. I know that was a really interesting paper for me to read, since my research is kind of focused on the same thing, structure-function relationship, and creating more structured products like steak, chicken breast, et cetera, like you're talking about.
Yeah.
So yeah, I guess I have a couple of questions kind of revolving around this transition between academia and starting a company or joining industry. Could you kind of describe that process? Like, when did you first realize you guys might want to start a company? How did you identify that you fit, you know, a niche that was needed? And yeah, we'll start with that.
Yeah, I mean, I think me and some of my co-founders, I think we were always interested in starting a company. Even me, from personal background, I wanted to go in something related to climate, and I was doing research in regenerative medicine, which is really interesting, but I kind of wanted to go back into that. So I think there was always kind of this personal interest from us to do that. I think when we realized that it was the right time for us, I think, you know, Memphis Meats and a couple other companies launched their companies in 2015, 16. We stayed quite a lot longer in academia, but when we published that paper in 2019, it generated a lot of interest, which I think for us was very exciting. But at the end of the day, it kind of boiled down to having, you know, a couple different things. And maybe for background, I think what's important to say is that this alternative protein industry is typically the companies that are launched typically require a lot of funding, a lot of capital to do research. And so going after venture capital investors was, in our opinion, like the better route for us. And yeah, so then for that, it was, you know, kind of aligning the different things that we needed to be able to kind of raise capital from these types of investors. And so we had a team. The couple different things that you typically need, you know, is having this core team, you know, that's competent at what we're doing. And I think that we kind of had. It's having the right opportunity. So we're going after this meat industry, which is gigantic, you know, and so if we're kind of narrowing it down to this kind of this, whether it's even scaffolding or texture, I think it's still a very large problem within this industry. And then it's actually having, you know, technology or a solution to go after that opportunity, that problem. And so we kind of realized kind of a little bit that we had some of those things. Obviously, if you have, you know, customer traction and revenue, those are all kind of additional kind of great things. Obviously, we didn't have those, but we have those kind of first three. And it, allowed us to kind of generate interest from investors, and then eventually raise some first money. And then, now we're probably going to raise a lot more, I think, just because things have been going well and faster than planned, and we're going to continue to allocate more resources to solving those problems.
Awesome. So you talked about kind of the funding side of things, and you said that initially you went after VC in the beginning. Mm-hmm. So I'm kind of curious, you mentioned this NSF grant, which you said I think was an SBIR mm-hmm. grant. Could you explain kind of the motivation behind that versus VC, and maybe some of the pros and cons of different avenues?
Yeah. So, I mean, we went after both. I think that the only thing is that, so government funding, you know, takes a while. So I think the review process was something like 7 or 8 months, maybe in total. So yeah, it's a lot of time. And so in the meantime, you know, we were talking to investors, and then we ended up getting money before. So that's kind of just how it happened. I think we were excited by both. The benefit of government funding is that it's non-dilutive, which is great, which is kind of free money. The downside is that, it is kind of weirdly enough, they tend to, there's more kind of rules and guidelines on what you can do, and like we have specific plan, you know, on what we're supposed to do with this money. Venture capital money, there is a plan, but it's it can be, I'd say, more flexible. So I think both are good. I think with the hindsight, I think we should probably have applied, you know, for NSF funding maybe 2 years ago. So we would have money, you know, to essentially leave the lab right away. It didn't kind of happen that way. Maybe the NSF was not ready to kind of fund that research anyways. I don't know. It seems like, the NSF grant that you received was kind of, maybe 6 months before ours or something like that. So it was maybe a timing thing also. But I think both can be good. It depends what you're looking for.
Yeah, that's a great distinction, and makes a lot more sense that it's kind of more structured, and non-dilutive is a good term that I just learned. So I guess one more question, and then we should jump over to Kathleen. You have this background in more regenerative medicine, and since this is a career fair, I'm sure we have a lot of students in the audience who don't have a background in cultivated meat or alternative protein. And like as the president of the Alternative Protein Project at Davis, I have a lot of students coming to me asking kind of how they can position themselves for industry.
Yeah.
Could you maybe share some thoughts on kind of different backgrounds and what you're looking for in candidates?
Yeah, I mean, what I think is really awesome about this industry is that we need people with a lot of different backgrounds. So if you just look at our website, I mean, we're hiring people that have biochemistry backgrounds, material science backgrounds, cell biology, mechanical engineering, because essentially, well, if we're talking about plant-based or cell-based, often like it's a whole industry that needs to be created, and so you need expertise in a lot of different things and at a lot of different levels. So we just went through hiring, I think, 5 junior people that have experiences in mechanical engineering, material science, bioengineering, and now we're going to hire some people in food science, in cell biology. So I think there's really a lot of different expertise that are needed. I think that's kind of the simple answer. And yeah, I mean, like us, we had backgrounds in regenerative medicine, but we're applying it now to this cultured meat industry. And if now, like there are actually people that can be trained in that industry more specifically, great. But otherwise, we'll take people that from other industries. I think that's I think our lead chemist, has worked in the paint industry previously, and just because that's where he was learning a lot of how polymer chemistry and all that stuff, and it was very relevant to what we're doing. So yeah, and yeah. I think, again, we hire at all levels, it's people that just graduated or people that have done PhDs and postdocs and even more. So I think there's room for everyone, which is great.
Awesome. Thanks. Yeah, that's kind of what I tell people as well is, we need so many different backgrounds and like the tissue engineering side is just one component of the equation, right? Once we get to all the different aspects, you need whole different backgrounds that really aren't necessarily immediately relevant. So if anyone's listening and they're not aware of it, there is actually this resource called Pathways from CellAg Australia. I think if you just Google that, you should be able to find it. And it discusses a little bit about some different kind of disciplines and majors and maybe how that's relevant to the industry and how you can position yourself. So that might be a good resource to check out. But thanks for talking, Christophe. It was awesome to hear about what you've done. And now we'll pass it off to Kathleen.
Okay, can you hear me?
Yep.
All right, let me see if I can share my screen. Is that coming out?
Not yet.
Oh, I have to click. Can you see my presentation?
Yeah. Yep,
okay.
We see it now.
All right. Okay, so thank you very much for inviting me. I'm going to talk about plant-made replacements for animal food proteins using technology known as molecular farming. And just a little bit of information about myself. I teach at Cornell University in the Department of Microbiology, and I am launching a startup company known as Forte Protein. All right. So molecular farming has actually been around for a while, for maybe 2 decades. And I've been one of the researchers in this field for quite a while. And originally, we were looking at making vaccines in plants. And this technology of molecular farming means that we're using biotechnology to produce pharmaceutical proteins in plant tissue. And so I've just given you on this slide, some reasons why this is kind of a worthwhile area of research to devote time to. First of all, millions of people in developing countries die from preventable infectious diseases every year. The vast majority of these are children under the age of 5. And often some traditional vaccines can be very difficult to get hold of or very expensive. And we know right now with the COVID pandemic and developing countries that we're seeing that as we speak. And what we found is that by producing pharmaceutical proteins such as vaccines or monoclonal antibodies or other therapeutic proteins, we can do this very inexpensively in plants. And just by increasing the number of plants, we can scale up very easily. And we can actually work without requiring refrigeration or trained medical personnel or syringes. So these are efficacious vaccines and pharmaceutical proteins that don't necessarily require, the same cold chain or, you know, um, uh, uh, sophisticated equipment that one needs for, other conventional pharmaceuticals. So I figured that this might sound a little science fiction, like science fiction to some people listening. So I'm giving you 3 examples of how plants have been used to make pharmaceuticals. And this first one is a company known as ProteinX Biotherapeutics. This is a company in Israel. And on the left-hand side, I'm showing you what are really 8-foot tall bags of carrot suspension cells. And these carrot cells are expressing this glucocerebrosidase, which is just an enzyme needed for treating Gaucher disease. Gaucher disease is a genetic disorder that some people are born with. And to get the proper conventional therapy for this, it costs about $100,000 US a year. But by using carrot cells, which will fold this complicated, kind of complex protein properly so that it functions well, we can make this protein for pennies a glass. So we can produce something that's actually drinkable and easy to manage and, very, very inexpensive. So this is one example. Another example you may have heard of was in the an earlier, I don't think it was the latest, but an earlier Ebola outbreak in West Africa. We actually had the issue of using a special monoclonal antibody cocktail that was produced from tobacco plants. So in this case, we used a plant expression vector, which was initially developed at Cornell University in the group I was involved in. And we made monoclonals to this particular virus. And it was the monoclonals were provided to some Americans who had contracted the disease while traveling in Western Africa. And it seemed to help them recover. So this is an example you may have heard about in the news. And then a third example I'm just providing you is that a large company known as Medicago in Canada is now producing a COVID vaccine, which is under its phase 3 trials and will be available very soon. And again, some of these plant-made vaccines are very easy to work with and don't require refrigeration. And here I'm showing you, instead of actually even ingesting this particular vaccine, we can even supply it as a patch, so it's intradermal. So that's easy to maneuver around in developing countries. So these are just 3 examples. Just to summarize this, I'm mentioning that we can change or improve global health using plant biotechnology. And not only can we, completely purify proteins or, pharmaceuticals and provide them to developing countries, as I'm showing on the top, we can also use plants and just take a crude extract of particular vaccine protein of interest and provide that to developing countries. So this is a area of research I've been involved in for quite a while, and it gave me the idea to move forward and start a company based on this technology called Forte Protein. So this is a molecular farming approach that we're using to make plant-based alternatives to animal food proteins. And I won't talk in much detail about the technology, except to say that it was developed at Cornell, and it's transient vector system that we're working with. And so we're interested in producing animal proteins sustainably and without using animals at all. So we're just using plants. It's quite easy. We have a lot of background, 20 years of experience in molecular farming. So it's, we're able to move quite quickly, I think, in this field. And I really like the idea from a broader perspective of making proteins more accessible and affordable for underserved or malnourished communities around the globe. So I think we could really make a difference for people. So these are just some of the problems that I think probably everyone would be familiar with, from dealing with increasing consumer demand for non-meat forms of or sources of protein, to being concerned about carbon footprints and greenhouse gas emissions, to even we have issues right now with global inequality and food security and protein deficiency. So these are all concerns going on in the world today, and are going to be concerns tomorrow as well. And so our process is that we can use a technology that in a way resembles a microbial bioreactor, except we use sunlight and soil and plants as our reactor. We can use a greenhouse or a vertical farm and expand very rapidly. There's a lot of different styles available for increasing plant biomass. And we can either purify our proteins, or we could use a partially purified protein, as I mentioned before. So using the greenhouse scenario, we do have the downstream processing, but our upstream processing we can scale up very rapidly and very cheaply. So our upstream process is really inexpensive as opposed to the microbial fermenter. So I think it's worthwhile to pursue this, certainly when thinking about underserviced communities, keeping that in mind. On this slide, I'm just showing you environmental impact comparison, and here I'm using some dairy proteins to give you kind of an idea that we can, because we're using this greenhouse or vertical farm, we really reduce our carbon footprint quite a bit. And so that's quite attractive from a sustainability point of view. And another thing, that because of this proprietary technology, we find that we can use our system on a variety of different food crops. So we have a lot of flexibility. We could work with soy or pea, or we can work with tomatoes or lettuce or kale. We have different prospects for producing these proteins in these different crops, if that is what is required. And I'll just end by saying that we're, we think that we really have a method here of improving sustainability and working towards the sustainable development goals. And, we'll see where this leads. So thank you very much for listening. I'm going to stop sharing if possible.
Thanks, Kathleen. That's really cool. This is actually a technology that I haven't been as familiar with lately. So this is really cool to see. I have a couple questions. So I guess with the company Forte Proteins, what is the target that you're aiming for? Are you looking to produce proteins to sell business to business, or are you looking to create full products?
That's a great question. So I think that initially it would be a B2B type of business. We seem to have, elicited some interest from other businesses who would like certain ingredient proteins that they either are having difficulty getting, the equivalent of in a, plant-based system, or it's too expensive for them to use for their purposes in microbial fermenters. So I think that would be our first shot. I think it would be fun to imagine moving forward and producing our own products in the future. So that would be kind of a later plan. And in fact, I think there are other startups at Cornell who we could probably collaborate in that regard as well, or maybe at Davis. So I think that there's a lot of room for interesting alliances and relationships.
Awesome. And it's awesome to hear that you're open to those collaborations and alliances as well. I know that's key kind of in driving this field forward. So what does the regulatory side of these types of products look like versus kind of more the precision fermentation products?
Yeah, so we're working towards figuring this out. Where we think that in general it will be a protein by a case by case basis, where we'll have to sort through what proteins we're looking at. But in general, it seems that a lot of these proteins could be considered generally recognized as safe. That's the impression we're getting with a lot of the ingredient proteins, so that they won't necessarily have to go through a lot of significant regulatory hurdles if they're considered to be dietary supplements or they have GRAS status. So that's kind of, we would probably be looking at the low-hanging fruit to begin with. That makes the most sense. But that's what we're being what's being presented to us so far. Thanks for all the likes.
Yeah, it's always fun seeing all these emojis fly up on the screen. Huh? So is this the first company that you have founded?
Yeah, that's a great question. So I actually had started another company just before the pandemic hit, and it was in Toronto, based in Toronto. And we had to put a kibosh on it because of the pandemic and my inability to travel across the border. It just became too difficult, and Toronto had a very strict lockdown as well, like much stricter than anything you would see in the United States. So, so out of frustration, I started thinking about starting something at Cornell. Why not? Since I was, we were all under lock, in a similar scenario with COVID there, and I was kind of welcomed into the fold. I would say I think I was given a very positive experience with the university in terms of this sounds like something we'd like to see move forward, and so it's been a very great and fun time. The company in Toronto was making specifically vaccines. I was very interested in vaccines, and this one is different. It's about food.
That's kind of ironic that it was such a timely company, but timing just didn't work out. It's pretty funny actually, but
It is
I'm
funny.
sure very frustrating for your side.
Yes.
So with Forte, are you guys, where are you in the growth cycle? Are you guys currently hiring, or what type of timeline should students expect?
Yes, we're actually right now looking at some interns. We have, we're starting to spread out a little bit in terms of hiring. Where we actually have 3 women founders, which is kind of fun too, to be founded by women. But we are starting to look towards hiring people. And my answer is very much the same as Christophe. It seems that we will be looking for all sorts of different disciplines, not just from the business side, but also from the scientific side. So, probably a food scientist would be very helpful. I think some engineers would be very helpful as well. So we'll be looking for, a variety of disciplines to help us move forward.
Awesome. And as a lecturer for many years, do you have any additional insights maybe to this transition between academia and industry, and maybe how the two can work together a little bit more?
That's really great. So I think universities all have their own IRB a board and conflict of interest management plans. And from what I can gather in my case, because I'm teaching something that's quite different, it's not really necessarily directly related to food. I'm teaching general microbiology, which right now we're talking. Tomorrow I'll be lecturing about the cell wall and gram stains. So there's not really a direct conflict of interest. So I don't have much of a management plan, but for other folks who might have a lab running that is working on a particular research topic or interest, and then have a startup company, one might have to set up a management plan with the university where, efforts are made to make sure that there's no outlying, serious conflicts of interest. And that can be, I actually know this from taking the Cornell IRB course. We have, they might ask you to do things such as uh, uh, um, have data analysis from another source, or just have other places to get some of the work done, so that you're not necessarily relying on your students who are trying to graduate to do your company's work. That's the kind of thing you want to avoid. And so there are strategies that a university will set in place to assist you, so that you don't fall down these rabbit holes, if that makes any sense. Unfortunately, by teaching microbiology, I'm not really quite falling into any rabbit holes at all, so I'm in a lucky place.
Yeah, that sounds like a nice position to be in.
Thank you.
So Christophe and Kathleen, thank you for sharing some content about your research and both of your companies. I know I caught wind of Boston Meats kind of right as you guys started, and it's been really awesome to see how much you've grown in the past, 8 months to a year or so. And I'm excited to hear about Forte and learn more about you all, and see your growth in the next year or so. I'm sure there are a lot of cool innovations on the horizon. Yeah, if you guys have any more insights to share or questions that I didn't ask, I think we have maybe a couple minutes left before tossing it back to Anna.
The one thing I'd add is if people are in the audience, students or things like that, and are interested by the industry, I'd really kind of I guess I suggest you guys start organizations, kind of like Nick did. We had a couple of students that did that at MIT, and they're starting like a cell ag organization, and maybe they're going to do a class next year. There's all of these schools have everything, to be able to support this kind of industry, but there isn't really kind of a framework in place sometimes. And so, just sometimes it's up to the students, to kind of force it in the right direction, maybe, whether it's, classes on that or on food science. My co-founder, Grant Gonzalez, did material science because there was no food science program at Harvard. And so material science was probably the closest thing to it. But yeah, I think it's student interest will drive a lot of it on that side. So if you are interested in this kind of stuff, I would strongly encourage you guys to do that. Yeah.
Yeah, and as Christophe said, as a PhD student right now, I'm the president of the Alt Protein Project at Davis. I also get to interface with the Good Food Institute and some of the other chapters of the Alt Protein Project, and some of the students at MIT, like Christophe was talking about. And there are a lot of students that are really driving a lot of change kind of throughout the university ecosystem. And it's really amazing to watch and be a part of. And we love your help. And if you have any questions of maybe how to get started or how to get connected, feel free to reach out to me. I'll be happy to help as my time allows. And yeah, I'll just toss it back to you, Anna.
Yeah, thank you guys. Thank you for taking the time. It was great to see, the work everything that you guys have been working on. So thank you for being here with us today. So with this mind-blowing session, we wrap up our today's presentation.